Now, it's been known for a number of years that neurons and glial cells release molecules that allow them to communicate with one another. But around 5 years ago, this paper came out in the journal Nature, which prompted the field to really start to rethink the mechanisms or processes that might underlie this communication between neurons and glial cells. And essentially what this paper showed was that in an Alzheimer's mouse model, if they used artificial methods to boost the levels of these waves of electrical activity, these gamma oscillations in their brain, what they also showed was that the properties of their microglia cells, remember these are the sentinels of their brain that coordinate and direct these immune responses, they also changed. So the cells went from having this star-like resting appearance to this more amoeboid, Pac-Man-like appearance in which they can go around and chomp up pathological aggregates. And remarkably, what they also observed in this paper was that when they boosted gamma oscillations and caused the microglia to take on this particular form, the overall amount of amyloid plaques that formed within the brains of these model mice decreased quite dramatically. And so this suggests that leveraging communication between neurons and glial cells and restoring normal patterns of activity or normal patterns of communication between these cell types could hold a key to treating Alzheimer's disease. And so one way that this group has taken, or one approach that this group has taken to do this is by using sensory stimulation in the form of light and sound to try and boost gamma oscillations in the brains of people with dementia. And so here we can see a member of this original research team wearing a device that provides light and sound stimulation to boost these gamma waves within the brain. But one thing that particularly interests me and my group is that we don't have a good handle on the mechanisms that underlie this process. And perhaps if we understand the molecular mechanisms that mediate this process, we could also target it with drugs to boost this process within the brains of people with dementia and thereby take an approach to treating the disease. Now, one of the difficulties that we have in trying to understand the mechanisms that underlie this process is that it's very difficult to get access to the brains of either people with dementia or even animal models. And so one of the things that we've been doing over the past year and a half or so is to develop a model in which we can study the molecular mechanisms that underlie this communication between neurons and glial cells mediated by these rhythmic patterns of brain activity. And we do this using slices of brain tissue. And we can place electrodes in this brain tissue, and we can also image this brain tissue and then use drugs to try and change the patterns of activity or block certain processes to understand how this works. And so one of the things that we observe in our model is that when there's an absence of this rhythmic brain activity, which is shown here, so this is a trace or a recording of electrical activity in this slice of brain tissue, what we see is that our microglia cells have this star-like resting appearance. So they're not in this sort of inflammatory state that can facilitate the clearance of protein-based pathologies. But what happens when we instate these rhythmic patterns of brain activity, these gamma oscillations in our brain slice, and so that's illustrated by this repeated pattern of oscillatory activity in our electrical trace. What we see is that our microglia change their appearance, so they take on this amoeboid-like morphology, and it therefore puts them in a state in which they're better able to engulf pathological protein-based deposits.